is Carla, and this is a picture of me. My topic today was on Alzheimer's disease, and the objective was trying to determine how or why the hippocampus is involved in Alzheimer's disease, as well as how the brain is affected by this disease. So what Alzheimer's disease is, it's a neurodivergent condition which is often characterized as an abnormal accumulation of amyloid plaque, neurofibrillary tangles, and neuronal loss. These are unusual clumps of protein found in the tissue between the nerve cells. So the articles and their objective I will be covering in the following slides, but this is just here a pretty much summarization of what they cover. Article number 1, the objective was determining lipid differences in Alzheimer's disease. It was a study on the human brain which showed lipid differences in the human hippocampus. And the method for this was performed using 12 frozen post-mortem human brain samples from the hippocampus block. 6 of them were normal and 6 were Alzheimer's disease brain samples. These samples were age and gender matched in each section, and the tool used to observe this was MALDI imaging, an MRI tool. The study revealed that there were both positively and negatively charged lipid ions in both normal and Alzheimer's disease tissue. However, it was still detected that the abundance of lipid distribution changed in Alzheimer's disease brains. Now, article number 2, the primary focus was to be able to figure out the expression levels of perilipins 1 through 5. This was specifically focused around the cerebral areas responsible for Alzheimer's disease, such as the temporal and frontal lobe. So what was used for this was 50 autopsy human brain samples around the ages of 33 to 104 years of age. They were evaluated by using Western blotting and confocal microscopy analysis. So what resulted out of this was that perilipin 2 appeared to be more expressed in gray matter, while perilipins 3 and 5 were expressed more in white matter. And this here is just an image of lipids, and to further explain them, lipids are fatty components that perform a variety of functions in your body. They're part of your cell membranes and help control what goes in and out of your cells. And this here is just an image of perilipins in the brain, 1 through 5. And to further explain them, they are conserved proteins that decorate intracellular lipid droplets and are essential for lipid metabolism. So continuing articles 3 and 4. So article number 3, its primary focus was the protective effects that ondisaponin B, I hope I pronounced that correctly, would have on Alzheimer's disease through the enhancement of the hippocampal neuroplasticity. This was done through a model of mouse. So this study used male mice who were about 6 to 8 months old, and the materials used were OB and modified silver staining. So the results of this was that it revealed that OB was the major effective bioactive component of Bredig polygala and showed very effective roles in helping the mice who struggled with Alzheimer's disease to cognitively function better. Article number 4, its primary focus was timing impairments and episodic memory in the hippocampus as levels of B-amyloid began to be elevated. This experiment used 24 male mice, 12 were wild-type mice, and 12 were control mice. The study was conducted using sucrose pellets and an infrared photobeam sensor to record. So what resulted out of this was that it was revealed that there appeared to be no genotype or change in learning or sign of trace conditioning deficit with elevated levels of B-amyloid. However, it was observed that the time pathology did in fact worsen with age. And this is just an image of the molecule of OB, as you see here, and this here is just an image of the elevated levels of B-amyloid and its phases. And to finally finish this off, here is just a list of my references.